Bismillah ar-Rahman ar-Rahim. Today I'm going to talk about a new type of cell death that was the focus of many recent researches. This subject is very interesting and very attractive in the scientific media, which lead to thousands of researches are done in these subjects in the recent years. Our lecture today is about NETosis. What's called NETosis? This paper that I found in published in Biochemistry in 2020, NETosis molecular mechanisms role in physiology and pathology, this paper was full of information that I needed 2 videos to trying to explain what comes in only this paper. Before going into details of the process of NETosis, I will give only a summary about the process. What is NETosis? NETosis is the program of formation of neutrophil extracellular traps. These traps consist of modified chromatin attached to it or decorated with bactericidal proteins that come from the granules and cytoplasm of neutrophils. So the process of NETosis means that the formation of extracellular traps from neutrophils that consist of modified chromatin, and this chromatin is decorated with bactericidal proteins from granules and cytoplasm of neutrophils. Induction of NETosis depends on the reactive oxygen species, which is formed as a result of activation of NADPH oxidase. How NADPH oxidase is activated? This one is activated through increase in the intracellular concentration of calcium, and also in some cases generation of reactive oxygen species from the mitochondria. This will activate NADPH oxidase, which will form reactive oxygen species that will result in the process of NETosis. The production of reactive oxygen species inside the cytoplasm of neutrophils will result in the release of granule components in the cytosol, and these components will go inside the nucleus and cause histone modification of histones, and will lead to chromatin decondensation and destruction of nuclear envelope, as well as formation of pores in the plasma membrane and the production of extracellular nets. This photo shows the extracellular traps on NETosis, extracellular traps from neutrophils. I think it's a nice picture of it. Neutrophils as a professional phagocytes contain many granules that are full of antimicrobial compounds. These antimicrobial compounds can destroy the pathogen in the process of phagocytosis. Not only this, these bactericidal enzymes can go also outside the cell and release during the process of degranulation and kill the pathogens. Neutrophils also have the capacity to kill bacteria through formation of toxic reactive oxygen radicals such as nitric oxide, superoxide anions, and hydrogen peroxide. This process is mediated by NADPH oxidase system, and frequently is named as respiratory burst, which is extremely potent and requires tight regulation to prevent a neutrophil autodestruction. Activation of NADPH oxidase enzyme leads to a mass generation of reactive oxygen species, which will result in destruction of the pathogens, either inside the phagosomes or outside the cells. The third mechanism that the neutrophil use to kill the pathogen is the process of formation of extracellular traps or nitosis, as I said, which consist of modified chromatin decorated with bactericidal proteins from the granules inside the cytoplasm. And because this process of formation of nets is accompanied by cell death, this process was called nitosis. The formation of extracellular nets or the process of nitosis is activated by various pathogens such as bacteria, fungi, viruses, protozoa, as well as bacterial cell wall components, lipopolysaccharides. Also, nitosis could be induced or activated by antibodies and immune complexes, cytokines and chemokines like interleukin-8, tumor necrosis factor, also activated by microcrystals like cholesterol and other physiological stimuli. Two main forms of nitosis was described. The classical or suicidal nitosis, that's the one I described just now, that leads to cell death. And the other type is vital nitosis, in which a cell retains not only viability but also many of its effector functions. The classical type of nitosis is already a special form of programmed cell death, which is characterized, as I said again, by release of granule components into the cytosol and the chromatin decondensation associated with histone modification, and then the formation of extracellular traps. And there are many shared characters between this type of cell death and other types of programmed cell death like apoptosis, necroptosis, pyroptosis, autophagy, and secondary necrosis also. So there are some shared features between them. Nitosis, unlike apoptosis, does not require activation of caspases, and also nitosis differ from apoptosis by being accompanied by disturbance in the insulating properties of the plasma membrane, while it shares the mechanisms of permeability changes with other forms of programmed cell death like pyroptosis and necroptosis. The signaling mechanisms of nitosis may include the activation of phosphoinositide 3 kinase, which also controls the autophagy induction. But here in nitosis, the process doesn't require autophagosome formation. The mitochondria also are involved in the transmission of signals that determine a response of neutrophils to pathogen and share in the activation of NADPH through mitochondrial reactive oxygen species, and also the mitochondria shares in the induction of nitosis caused by various stimuli. So mitochondria plays an important role in the process of nitosis. Nets formation has also been shown in other types of cells, of the immune cells, not only neutrophils. For example, from eosinophils and mast cells, basophils, monocytes, and macrophages. Actually, the main mechanisms of NADPH oxidase activation during the process of nitosis is not that much different from the activation of NADPH in the process of oxidative burst of neutrophils in response to pathogens and other stimuli. The actual mechanism of NADPH oxidase activation is through phosphorylation of its subunits by protein kinase C, which leads to activation of NADPH oxidase enzyme. Here is a diagram to show in detail the process of NETosis, which depends mainly in the production of reactive oxygen species or what's called oxidative bursts that come from activation of NADPH oxidase enzyme. And these reactive oxygen species will affect the nucleus to make chromatin decondensation and release of chromatin, and also will destruct the granules inside the neutrophils. How we activate this NADPH oxidase enzyme to generate reactive oxygen species is through protein kinase C, which will activate NADPH. And protein kinase C is activated through many ways, as we see in the diagram. One of them is important, the A23-18-7. A23-18-7 is a calcium ionoform, which means a mobile ion carrier that forms stable complexes with divalent cations like calcium. This one will mobilize the calcium from inside the endoplasmic reticulum. So A23-18-7 will lead to transferring of calcium from endoplasmic reticulum to the outside, and this will result in what's called crack or calcium release activated channels to be activated. These are specialized plasma membrane calcium channels which will be activated when there is low calcium inside the endoplasmic reticulum. This channel will be activated and opening and resulting in entrance of calcium from extracellular to inside the cells when it is activated. As a result, calcium concentration inside the cell will increase, and this will lead to increasing in the mitochondrial non-selective pores, activation of mitochondrial non-selective pores, and also formation of mitochondrial reactive oxygen species, which again will activate the non-specific mitochondrial pores. These mitochondrial reactive oxygen species also will go to activate the protein kinase C that will activate NADPH oxidase enzyme. There is also other ways to activate protein kinase C to activate the NADPH oxidase enzyme. One of them is through what's called PMA or the furbol ester, and another way through what's called FMLP for N-formyl ethanoyl leucyl phenylalanine. Okay, this will attach to certain receptor, and by complicated process will result in activation of protein kinase C and activation of NADPH oxidase and making this reactive oxygen species production. Another issue is that as we see, the mitochondrial reactive oxygen species that activate NADPH oxidase is oftenly referred to as the second mechanism, that is NADPH independent oxidase independent NETosis, but it's better to call it mitochondrial dependent NETosis. So to continue the process of NETosis, now we have reactive oxygen species overproduction. These reactive oxygen species will open or destruct the granules inside the neutrophil cell, So we have azorophyll granules inside the neutrophils. These azorophyll granules contain a protein complex called azorosome. This azorosome includes 8 types of proteins. 3 of them are highly homologous serine proteases that include neutrophilic elastases, cathepsin G, and azorosidine. And also these granules contain myeloperoxidase. The contents of these granules will go into the cytoplasm, and this serine proteases, which is primary neutrophilic elastase, will break down the cytoskeletal elements and contributing to the process of nitosis. After release of the contents of the granules, these enzymes will migrate to the nucleus, where they break down the lamine and histones, contributing to chromatin decondensation, and also result in destruction of nuclear envelope. Myeloperoxidase also plays an important role in the dissolution of azorosomes itself and release of proteases from the granules. An important player in the process of nitosis is what's called peptidyl arginine deaminase 4, or PAT4. This PAT4 comes from the cytoplasm to the nucleus to catalyze a process called citrullination of the histones, or histone enzymatic conversion of arginine residua to citrulline. And this will lead to chromatin relaxation. So this process is very important in the process of nitosis, and this is the one responsible for chromatin decondensation. And also in trials, inhibition of PAT4 resulted in inhibition of nitosis and chromatin decondensation. Okay, so this PAT4 is responsible for what's called histone citrullination, or enzymatic conversion of arginine residue into citrulline to let the chromatin relax and do chromatin decondensation. PAT4, or peptidyl arginine deaminases, are calcium binding proteins, and their activity is stimulated by calcium. And also PAT4 is activated and do its job as histone citrullination in response to addition of hydrogen peroxide. Another issue is that by experiments, lipopolysaccharides related citrullination of histones and nitosis depend on the intactness of the microtubules. So apparently combination of all these factors are necessary for activation of PAT4. There was also a mention about another process different from citrullination, which is acetylation during nitosis, but this process is still poorly understood. Chromatin decondensation, as well as proteolytic damage of the nuclear lamina by the proteases that came from the neutrophilic granules, this all will lead to destruction of the nuclear envelope and the release of chromatin into the cytoplasm and then outside. Recently, there was also mention to activation of cyclin-dependent kinase. This cyclin-dependent kinase is a player in the process of entering the cell into cell cycle process. Actually, it appears as that mitosis apparatus such as phosphorylation and centromere separation are used to help in the process of destructing nuclear envelope during process of nitosis. The final stage of NETosis, pores will be formed in the plasma membrane, and chromatin is released into the environment with nets formation, and proteins released from the granules will be strongly attached or bound to the decondensed chromatin due to electrostatic interaction. The pores in the plasma membrane that will allow these giant complexes to pass through it to the outside are formed by what's called gas-dermine D protein. Gas-dermine D protein, which also forms pores in the macrophages membrane during pyroptosis. However, this gas-dermine D protein was formed by cleavage or usage of caspases in the process of pyroptosis, while in our situation in NETosis, it's cleaved mainly by neutrophil elastase. Activation of gas-dermine D probably will lead to the formation of pores not only in the plasma membrane but also in the nuclear membrane. There is another one which is called gas-dermine E. This gas-dermine E is activated by caspase 3 and will lead to increased permeability of the mitochondria and promotion of apoptosis as well as secondary necrosis. Another pore forming protein is called MLKL. This one is not related to gas-dermine, and activation of MLKL in neutrophils can also lead to release of nets outside to the environment. On the other side, pathogenic microorganisms will not stay silent from these extracellular traps or nets, and they have already a wide range of tools that interfere with the microbial action of nets. Many microorganisms produce lytic enzymes, primarily endonucleases, that destroys these nets, and bacteria such as Pseudomonas, Mycobacterium TB, as well as fungi produce protective and masking extracellular envelopes as part of a defense mechanism. In other bacterial species, coating with sialic acid induces production of interleukin-10, which will suppress NETosis, process of NETosis, and also same mechanism is used by HIV virus, human immunodeficiency virus, through stimulation of production of interleukin-10 by dendritic cells, which will protect the virus from the lytic action of NETs enzymes. In other situations, like in hepatitis B virus, it will inhibit NETosis by suppressing the production of reactive oxygen species in neutrophils using the hepatitis B envelope protein and hepatitis B C core protein. So the process that I just described was about the classical NETosis or suicidal NETosis, the first main type. We have another type which is called vital NETosis, and in this vital NETosis we have DNA release from neutrophils, while neutrophils retain their viability and natural effector functions. However, the Cell Death Nomenclature Committee in 2018 didn't recommend to use the term NETosis for the process not associated with cell death. They want that to use the process of NETosis only when we have a cell death. An example of this vital NETosis is release of chromatin that was observed in vivo when the skin was infected with gram-positive bacteria. Interestingly, after NETosis, nuclear-free neutrophils were doing were active in doing chemotaxis and phagocytosis of bacteria. They found also a massive and very fast release of mitochondrial DNA without loss of vitality in eosinophils and neutrophils, primed with pro-inflammatory cytokines. For eosinophils, interleukin 5, interferon gamma, or granulocyte monocyte colony stimulating factor as regard the neutrophils, and stimulated with lipopolysaccharide. In post types of granulocytes, this process was dependent on the activity of NADPH oxidase, and a similar phenomenon also observed in activation of basophils. Recently also they found that the vital mitochondrial DNA release was found in B and T cell lymphocyte as well as in natural killer cells in response to oligodeoxynucleotides. And also we have to remember that extracellular mitochondrial DNA, especially the oxidized one, is found in the blood in a wide range of pathologies that include systemic lupus erythematosus and other autoimmune diseases. And so NETosis plays an important role in autoimmune disease. I think that's enough for the first video of NETosis, and we will continue the remaining of the paper in the other NETosis video. And before I close, I want to remind you to subscribe to the channel if you didn't, and to activate the alarm bell so that you can be updated with any new video. And thank you.